We're going to wrap up this chapter by talking about chromosomes. So we've been talking about cell division and reproduction, and we've been throwing around all these terms about chromosomes. We haven't talked a lot about chromosomes themselves. So before we move on, let's talk about chromosomes. Most of our chromosomes are autosomes. These are our non-sex chromosomes. In humans, we have 22 autosomes. So remember that we have 22 types of autosomes, but we have 2 copies of each. So we actually have 44 autosomal chromosomes. So we have 2 copies of each type. The remaining set of chromosomes are our sex chromosomes. Now they're called sex chromosomes because they determine the biological sex of an organism. In humans, males have an X and a Y chromosome. Females have 2 X chromosomes. Now, before we go on, I want to talk about some characteristics of chromosomes. So here we have our X and Y chromosomes. You can see that they're quite different. These X and Y chromosomes are actually considered homologous. But let's say that we're talking about chromosome number 1. In chromosome 1, we get 1 copy from mom and 1 copy from dad. Now, whether we're talking about an X, Y, or other chromosome, each of our chromosomes is characterized by the length of their arms and the position of the centromere. So you can see that the X chromosome has relatively long arms compared to the Y chromosomes. It does have 1 longer arm, and the X chromosome is actually one of the larger chromosomes. The Y chromosome is one of the smaller ones. You can see that it has a very small arm on one end, and then a slightly larger but still overall smaller arm on the other end. So if we look at our X chromosomes here, we're actually in our sister chromatid formation. And same thing here with our Y chromosome, sister chromatids. Down here, I have drawn homologous chromosome 1, but we are not in our sister chromatid form. So this would be 1 from mom and 1 from dad. If you've seen pictures of chromosomes before, you are probably most familiar with this X-shaped form. That X-shaped form has nothing to do with the X chromosome. That's just how sister chromatids look when they're condensed, ready for cell division. All right, so how do we actually determine sex in humans? Well, the Y chromosome actually determines whether or not an individual is male. Females have 2 copies of the X chromosome. Males have only 1 copy of the X chromosome, but instead they have the Y chromosome. The Y chromosome is unique in that it is not actually necessary for the development of a functioning human, which is why females are totally normal without a Y chromosome. Because of this X chromosome in males, they have all of the information that is required for a normal functioning individual. However, the Y chromosome has some unique genes that are not found anywhere in the X chromosome. The Y chromosome mainly contains genes that are critical for male sexual development. So these are genes that are going to encode male sexual characteristics, male genitalia, and genes for development of testes, which is included in genitalia and sperm. So the presence of the Y chromosome adds those additional genes that allow for the development of male sexual characteristics. During meiosis, the X and Y chromosomes are split up when homologous chromosomes are separated in meiosis 1. And so each gamete gets one copy of a sex chromosome. Now, if you're a female undergoing oogenesis, that is production of your eggs, you are going to pass on an X chromosome to each of your cells. If you are a male producing sperm, spermatogenesis, each of your cells that you produce is either going to get an X or a Y. Then when fertilization occurs, you have one of the eggs and one of the sperm. If the fertilized egg ends up with 2 X chromosomes, you develop into a female. An X and a Y, you develop into a male. Now, moving on from sex chromosomes, sometimes during meiosis, we actually have an error that occurs where chromosomes do not separate correctly. This is a process or a condition called nondisjunction. Nondisjunction leads to an abnormal or altered chromosome number. Basically, it's an unequal distribution of chromosomes during cell division, where a gamete can end up with an extra or a lost chromosome. And then when that gamete is fertilized by or fertilizes another gamete, the resulting organism has one extra or one less chromosome. Now, this is a problem because our bodies don't deal with extra or missing information very well. In fact, missing chromosomes are basically not survivable. There are no known instances of survivable monosomy. Monosomy means that the cell is missing a chromosome. There are some cases where an extra chromosome is found in a gamete, and those tend to be more survivable. Our cells tend to survive better with extra information rather than missing information. So let's talk about what happens in nondisjunction. So in nondisjunction, chromosomes fail to separate correctly. So here we have an example. We go through metaphase. Our homologous chromosomes line up in their tetrads at the metaphase plate. And due to the chromosomes not separating correctly, too many are pulled to one side and none are pulled to the other. The result, if we follow through metaphase 2 to the end, is that one gamete has ended up, or one half of the gametes has ended up with extra information. The other half of the gametes has ended up with missing. So they don't have, I only needed one of those. They don't have that green chromosome. They're missing that chromosome. Chromosome's not there. So instead of having one copy each of a blue and a green, they only have a blue. This is called a monosomy. The cells with the extra chromosomes are called trisomies, meaning we have 3 copies eventually in the fertilized egg of one type of chromosome. Because when this cell is fertilized by a normal, let's say this is an egg and it's fertilized by a normal sperm with 1 green copy, it's going to end up with 3 green copies. If this cell is fertilized by a normal sperm, it's going to end up with its correct blue copies, but then only 1 green, which is why we call it a monosomy. So trisomies, where we end up with 3 copies of one type of chromosome, are not common, but they do occur more than monosomies do. The most common one that you've probably heard of is trisomy 21, or Down syndrome. These trisomies are often associated with increased reproductive age, and we'll talk about why that is in just a second. So nondisjunction for chromosome 21, otherwise known as Down syndrome, is the most common trisomy. And the reason for that is that it occurs in one of our smallest chromosomes. So this is chromosome 21. Chromosome 21 is a really small chromosome. And the reason that this one is more common than some of the others is that the chromosome is so small. There's actually a small amount of extra genetic information. So a small number of genes are affected by multiple copies. It's relatively common for a non-disjunction disorder. It occurs in about 1 in 1,000 children. You guys are probably familiar with the characteristics of Down syndrome. I'm actually not going to really go into them here because for us, we're discussing chromosomes. But you are welcome to look up in your book if you're interested in the phenotype of Down syndrome. Before we move on, I want to note that this is a karyotype. A karyotype is basically a picture of chromosomes. So these are actually sister chromatids here. This is a homologous pair. And if you were to count a normal karyotype, you would see that there are 2 copies of each type of chromosome. So this is chromosome 1, 2, 3, etc. They are lined up by size. Down here at the bottom, we have an X and a Y. So this is a male karyotype. They are stained so that you can see the characteristics. I know you guys are probably not experts in reading karyotypes. You'll just have to take my word for it that they're stained to show that they are the same type. This karyotype is important because it does show that extra chromosome 21. So here's another example of a karyotype. You can better see the staining here to show that they are the same. Homologous chromosomes here, sister chromatids here. The staining reveals that we have the same physical characteristics. That's how we know we have 2 copies of chromosome 1, because remember, the genes are the same all along. Likewise, the characteristics are the same all along. So the staining pattern in chromosome 1 is the same for both homologous pairs. Down syndrome can be detected before birth using a karyotype. So this is a process. Cells would be extracted from the fetus or the amniotic fluid and processed into a karyotype and analyzed for the presence of chromosome abnormalities. So here, this karyotype is actually normal for trisomy 21. This is a female. We can see because she has 2 X chromosomes. Now, Down syndrome actually increases with the mother's age. It is thought to be due to a breakdown in the process of meiosis. Essentially, all of the eggs that are ever going to be produced by a female are made when she's a fetus, but the process of meiosis is interrupted. And so they believe that the reason for an increase in Down syndrome with maternal age is basically that those spindle fibers in meiosis break down between the time that she is a fetus and her eggs are produced and the late maternal age that the egg actually finishes meiosis and is used and fertilized to make a child. So you can see about age 35, between 35 and 40, the chance of Down syndrome starts to go up very quickly, and it increases at an exponential rate after that. So risk of Down syndrome is relatively low between, say, puberty and 35, and then begins to increase as the woman gets older. So Down syndrome is an autosomal trisomy, but we can also have sex chromosome abnormalities. So we can actually have an abnormal number of sex chromosomes as well, and they produce very specific syndromes as well. A non-disjunction event that results in the loss of an X chromosome is called Turner syndrome. Turner syndrome is the only survivable monosomy, meaning that we're missing a pair of the homologous chromosomes. So individuals can actually survive with only one X chromosome. It's the only chromosome where you can survive with only one. In general, individuals, females with Turner syndrome, lead normal lives. There are some characteristics of the syndrome, including a shorter height. You can see here there's some web skin along the neck and shoulders. They're often sterile because their sexual organs do not develop normally, and some learning difficulties. Klinefelter is another disorder that results from non-disjunction, and this is where the individual has an extra sex chromosome. So in this case, we have either a Y that is extra, or one of the X chromosomes is extra. In either case, these individuals generally have a male appearance, but they have underdeveloped testes. Sometimes they develop some female features, and then they have some other characteristic traits of this disorder, or this syndrome, actually. Klinefelter's is a trisomy, where you have an extra or 3 copies of one type of chromosome in the cell. So as we can see with Down syndrome, Turner syndrome, and Klinefelter's, you can actually survive and lead a relatively full life with chromosome abnormalities, although they do present some problems, and that's because our bodies don't deal well with missing or extra genetic information.